Hello students, welcome you all to a new video of Sunanda's tutorial. So today in this video we will discuss about the post-transcriptional modification in case of eukaryotes. In case of prokaryotes, they completely lack post-transcriptional modifications. Their transcription and translation occurs simultaneously. Okay, but here in case of eukaryotes, they have some post transcriptional modifications which takes place after the transcription process or along with the transcription process. So it is a series of biological process in which newly synthesized primary RNA transcript or pre-mRNA or heterogeneous RNA is chemically altered to become a matured and functional mRNA molecule. Okay. In case of eukaryotic cell, this occurs entirely within the nucleus before the mature RNA can be exported to the cytoplasm for translation into proteins. So the main mRNA which is formed after transcription is called as pre-mRNA or hnRNA. hnRNA means heterogeneous nuclear RNA. Okay. In case of eukaryotes, the transcription process takes place inside the nucleus and the translation process takes place in cytoplasm. Okay. So mRNA after processing and modification exit the nucleus and enters inside the cytoplasm where the translation takes place and protein is formed from mRNA or matured mRNA. Okay. Prokaryotes generally do not undergo this processing because transcription and translation happen simultaneously in the same compartment in case of prokaryotes. Okay. So what are the processing or modifications? At first, 5' capping, 3' polyadenylation, RNA splicing. So these three important modifications takes place after the pre-mRNA is formed by the process of transcription in eukaryotes. Okay, so after the transcription in eukaryotes, the pre-mRNA or immature RNA or hnRNA will undergo 5' capping or 3' polyadenylation and RNA splicing. Okay. So at first, what is 5' capping? 5' means 5' end capping. A cap is attached to the 5' end of the newly formed mRNA after transcription. Okay. Which cap? It is 7-methyl guanosine cap. It is added to the 5' end of growing RNA chain after transcription slightly begins. This cap protects the transcript or mRNA from rapid enzymatic degradation. So what is its role? It protects the 5' end of pre-mRNA. It protects the 5' end of pre-mRNA from enzymatic degradation by exonuclease. So what is the role of exonuclease? It can cleave the RNA or DNA from the end, but endonuclease can cleave the DNA or RNA from any part. So this cap assists in nuclear export and serves as vital recognition signal for ribosome binding during the initiation of translation. So it is very necessary. So now what are the steps of 5' capping? So remember, the first important step is dephosphorylation. That means phosphate group is detached from the main mRNA. Second, guanylation. Guanylation Okay. Third point is methylation, addition of methyl group, methylation. Okay. Dephosphorylation. At first, dephosphorylation, the an RNA triphosphatase enzyme removes the gamma phosphate group from the 5' end of the new RNA strand. Okay. So after the removal of one phosphate group, two phosphate groups are attached to the 5' end, diphosphate. Next step is guanylylation, addition of guanine. A guanylyl transferase enzyme, guanylyl transferase enzyme adds GTP molecule to that RNA diphosphate end, creating a special reverse 5' to 5' triphosphate link. Next step is methylation. Methylation means addition of methyl group. Methyl transferase enzyme adds methyl group to the 7' position of new guanine cap. Okay, so in this way the 5' capping takes place to the newly formed mRNA. Okay. Next step is polyadenylation. Polyadenylation, that means the addition of a poly A tail. A tail means adenine tail at 3' end. So at 5' end a cap is attached. At 3' end the transcript is cleaved at a specific signaling sequence AAU. An enzyme called poly A polymerase adds a long chain of roughly about 100 to 250 adenine nucleotides. And this poly A tail detects mRNA stability, regulates the molecule's lifespan in the cytoplasm, and it facilitates efficient translation machinery recruitment. So presence of this poly A tail helps in the stability of a newly formed mRNA. It regulates the molecule's lifespan in the cytoplasm. It also facilitates an efficient translation process and translation machinery recruitment. Okay. Next, what are the steps of poly A tail or polyadenylation? At first, recognition. Specific protein complex including cleavage and polyadenylation specificity factor CPSF, cleavage stimulation factor CSTF, they bind to conserved sequence elements on the newly transcribed pre-mRNA. Most notably the polyadenylation signal. Okay. Then cleavage. An endonuclease cleaves the mRNA transcript at specific site downstream of the recognition signal. Next, the polyadenylation. Poly A polymerase binds to the newly formed 3' end and add 50 to 250 on templated adenosine residues using ATP as substrate. So finally, a poly A tail at 3' end is attached. Okay. So this is the diagrammatic representation how poly A tail is attached to the 3' end. Okay. Now RNA splicing. Eukaryotic pre-mRNA contains both protein coding region exons and non-coding region called introns. A massive molecular complex consisting of small nuclear ribonucleoprotein snRNPs and auxiliary proteins collectively known as spliceosome precisely coat introns and paste exons together. So by the process of RNA splicing, the introns are cleaved from the pre-mRNA and exons, only the coding part of mRNA, is attached with each other. Okay. The spliceosome machinery, the majority, what are the spliceosome machinery? What are the tools? The majority of nuclear pre-mRNA splicing is catalyzed by a molecular machine called spliceosome. which is composed of small nuclear ribonucleoproteins and labeled as U1, U2, U4, U5, U6 along with hundreds of helper proteins. So all together are called as spliceosome. Okay. Spliceosome coordinates the mechanism through tightly regulated assembly sequence. So at first U1 snRNP bind to 5' splice site, while the helper protein bind to the branch point and polypyrimidine track. U2 bind to branch point sequence, causing target adenine residue to bulge out, expose its reactive 2' OH group. Then U4, U6 and U5 tri-snRNPs complex joins the assembly, bending the pre-mRNA to bring the 5' splice site and 3' splice site and branch point physically close to each other. U1 and U4 are ejected, a conformational rearrangement that activates the catalytic core formed by U2 and U6. So this is how they cleave the introns from the exons between two exons. Okay, and this is how the RNA splicing takes place for cleaving the introns from which are present in between exons. Okay, so these are major steps of post transcriptional or core transcriptional modification in case of eukaryotes. With this, I am concluding my lecture video here about the post transcriptional modification, but I will discuss the RNA splicing, particularly RNA splicing, in a separate video. Okay, so please stay tuned, and if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.